Welcome, curious minds, to the deep dive. Today we're embarking on really a fascinating journey into one of the most intricate and vital systems in your body, the endocrine system.
It really is fundamental.
Yeah, and our mission, if you will, is to unlock the profound secrets of how these chemical messengers are made, how they, travel throughout your body, and the incredibly precise ways your cells recognize and respond to them.
It's all about communication.
Exactly. Our goal for you today, our dedicated listener, is to give you a foundational, almost mastery-level understanding of endocrine hormone action, their production, transport, and storage. Think of it as gaining the kind of conceptual insight that, elite professionals in this field possess, understanding not just the facts, but the interconnected elegance of it all.
Right, the why behind it.
Precisely. At its core, the endocrine system, alongside the nervous system, is all about intracellular communication. It's how multicellular organisms like us adapt to a constantly changing environment, reacting to both subtle shifts and, well, dramatic challenges.
And what's truly remarkable here is how these two communication systems, once viewed as pretty distinct, the nervous system as a fixed wired network and the endocrine system as mobile messages sent through the bloodstream, how they've converged in really surprising ways.
Oh, how so?
Well, we now understand that the nervous system plays a crucial role in regulating many hormones. Lots of neurotransmitters, the brain's chemical signals, strikingly resemble hormones in how they're made and how they act.
Ah, okay.
And in a twist, some hormones are even synthesized right there within the nervous system itself. It really speaks to a deep, shared evolutionary heritage.
Wow.
The very word hormone actually comes from a Greek term meaning to arouse to activity, which I think perfectly captures their dynamic role in getting things done.
That's a perfect description. And when we talk about hormones, the classical definition often comes to mind.
Yeah.
A substance made in one organ, then transported by the bloodstream to act on a different, often distant target tissue.
Yeah, that's the standard textbook one.
That's a good starting point, but it turns out to be, well, far too limited.
Exactly. That classical definition, while historically important, it just doesn't capture the full picture of how hormones operate.
Right.
It raises an important question. Why is it too restrictive? Because hormones can also exert their influence locally. They can act on adjacent cells, a process called paracrine action, without ever entering the general circulation.
So like cells chatting with their next-door neighbors.
Precisely. Think of cells in a specific tissue communicating directly with their neighbors. And even more directly, a hormone can act on the very cell that produced it that's known as autocrine action.
Ah, self-regulation.
It's a form of self-regulation, exactly. This incredible diversity in how hormones are made, stored, released, transported, and broken down is what allows for the exquisite precision and adaptability we see in our body's homeostatic responses. It's really a testament to millions of years of evolutionary fine-tuning.
It's incredible. And to give you this comprehensive picture, this deep dive is meticulously pulled from a foundational text, a classic in the field. HARPER's Illustrated Biochemistry, specifically Chapter 41, The Diversity of the Endocrine System, authored by P. Anthony Weil, Ph.D.
A fantastic resource.
Absolutely. We've combed through it to extract the most important nuggets, ensuring you get comprehensive insights directly from a top-tier source. So let's begin decoding these secrets, not just by reciting facts, but by truly understanding the underlying logic. Okay, let's unpack this with the fundamental principles of hormone action. It all begins with understanding how a hormone, this chemical messenger, even begins its journey, and crucially, how cells respond. Hashtag tag A, defining the hormone.
As you mentioned, the classical definition describes a hormone as a substance synthesized in one gland or organ and then transported through the circulatory system to exert its effects on a distinct, often distant, target tissue.
Right, the long-distance call.
Exactly. This is the traditional endocrine pathway, often referred to as endocrine action. Think of insulin from your pancreas traveling through your bloodstream to tell muscle and fat cells to absorb glucose, or thyroid hormones from your neck influencing metabolism across your entire body. They're long-distance communicators.
But, as we hinted, that classical view is too narrow. The body has evolved more immediate ways for hormones to act.
Precisely. Beyond the classical endocrine action, we have paracrine action. Here, a hormone is secreted by a cell and then acts on nearby cells, often within the same tissue or organ, without needing to enter the general circulation.
So the local gossip network.
You could put it that way. It's like a whispered message passed between close neighbors, rather than a broadcast to the whole town. A great example is how cells within the pancreas communicate with each other. Insulin-secreting beta cells can influence glucagon-secreting alpha cells nearby.
Okay, that makes sense. And then there's autocrine action, which is even more localized, perhaps the most direct form of hormonal communication.
Right. A cell synthesizes and releases a hormone, and that very same hormone then acts back on the cell that produced it.
Creating a feedback loop right there.
Exactly, a self-regulating feedback loop at the cellular level. This is crucial for processes like cell growth, differentiation, and aspects of immune responses. Some growth factors, for instance, can stimulate the proliferation of the very cell that secreted them.
Wow.
The profound implication here is that the body employs this diverse toolkit of hormonal communication modes. This evolutionary diversification in their biosynthesis, storage, secretion, transport, and metabolism is what allows for such an incredibly wide array of precise and robust homeostatic responses.
So each mode offers something different: speed, scale.
Exactly. Scale and speed of communication, essential for maintaining the body's delicate balance in a constantly changing internal and external environment. This biochemical diversity isn't just an interesting fact, it's absolutely central to our survival. #B, the crucial target cell concept.
So if hormones are these incredibly varied messengers, how do cells know which messages are for them? That brings us to the crucial concept of the target cell.
Yes, fundamental.
When you think about the sheer scale in the human body, it's truly mind-boggling. We have over 200 differentiated cell types, yet only a handful of those actually produce hormones. But here's the kicker. Virtually all of your approximately 75 trillion cells are targets of one or more of the 50-plus known hormones.
It's an immense network.
It's an information highway, and every cell is on it, listening for its specific instructions.
That's right. The concept of the target cell is absolutely fundamental. Historically, we had a simpler idea. We thought a hormone affected a single cell type, or maybe just a few, eliciting a unique and predictable biochemical or physiological action.
Kind of one-to-one thing.
A relatively straightforward, almost linear view of hormone action, yes.
But the modern understanding is far more complex and, frankly, much more reflective of the dynamic nature of biology.
Much more nuanced.
We now know that a single hormone can affect several different cell types, sometimes in surprisingly different ways depending on the cell. Conversely, more than one hormone can affect a given cell type, leading to intricate interactions and crosstalk.
Complex signaling webs.
And to add another layer of complexity, hormones can exert many different effects within the same cell or across various cells. It's not a simple one-to-one relationship. It's a dynamic, multi-layered communication network.
Precisely. And with the discovery of specific cell surface and intracellular hormone receptors, the dedicated listening posts of the cell, the definition of a target cell, has expanded significantly.
Right, it's about the receptor now.
It now includes any cell where the hormone, the ligand, binds to its receptor, even if we haven't yet fully pinpointed a biochemical or physiological response. The very act of specific binding is the defining characteristic of a target cell. This allows us to map out the full reach of a hormone's influence.
So this is where it gets really interesting. What actually determines the response of a target cell to a hormone? It's not just about the hormone being present, it's a symphony of factors.
It really is.
We can categorize this into two broad areas, right? Factors that influence the hormone's concentration at the target cell, and then factors that influence the cell's actual response to that hormone once it arrives.
Exactly. Starting with the factors affecting the concentration of the hormone at the target cell. The first and most obvious is the rate of synthesis and secretion of the hormone by its producing gland.
Makes sense. How much is being made?
If the hormone isn't being made or released sufficiently, its concentration at the target cell will inevitably be lower. It's the supply side of the equation.
Then there's the proximity of the target cell to the hormone source. This brings back our earlier discussion of endocrine, paracrine, and autocrine actions.
Right. A hormone acting paracrinely on an adjacent cell will be far more concentrated than one that has to travel through your entire circulatory system to a distant target, where it gets diluted significantly.
Distance matters.
Absolutely. The farther it has to travel, the more it disperses.
Okay.
And crucially, for many hormones, especially the fat-soluble ones, we consider the affinity of the hormone with specific plasma transport proteins. These proteins bind to hormones in the bloodstream, essentially acting as carriers.
Like little taxis for the hormones.
Kind of, yeah. If a hormone is tightly bound to a transport protein, less of it will be in the free, active form available to diffuse into target cells. It's a dynamic equilibrium, where the bound form acts as a reservoir and only the unbound form can signal.
So the binding tightness is key.
Very key. The fourth factor is the conversion of inactive or suboptimally active forms of the hormone into the fully active form. Some hormones are released as prohormones, needing a final enzymatic tweak to become fully potent.
Can you give an example of that?
A prime example is testosterone, which is often converted to dihydrotestosterone, DHT, in target tissues like the prostate. DHT is actually the more active form for many of testosterone's effects.
Ah, so it gets activated locally.
Exactly. If this conversion pathway is impaired, the target cell's response will be altered, even if the initial hormone is abundant. It's a clever way to control activity at the local level.
Very clever.
Sure.
And the last one for concentration.
Finally, we must consider the rate of clearance of the hormone from plasma. This involves how quickly it's removed by other tissues, degraded by enzymes, metabolized, or excreted by the kidneys or liver.
How fast it gets cleaned up.
Right. A hormone with a rapid clearance rate will have a shorter half-life in the bloodstream, and thus a lower effective concentration at its target cells over time. Understanding this delicate balance between production, transport, modification, and degradation is absolutely crucial for appreciating how hormone levels are precisely managed.
Now let's shift to the factors that determine the actual response of the target cell to the hormone, even if the concentration of the cell is perfect. This is where the cell's internal machinery comes into play.
Yes, the cell side of the equation.
First, we have the number, relative activity, and state of occupancy of specific receptors. Think of receptors as the cell's listening devices, or locks.
Good analogy.
If a cell doesn't have enough of the right kind of locks, or if those locks are faulty, or if they're already jammed by something else, then the hormone, no matter how abundant, can't exert its full effect. The density and health of these receptors are paramount.
Indeed. The metabolism of the hormone within the target cell itself is another key factor. Some target cells are not just passive recipients. They possess enzymes that can further activate or inactivate the hormone once it enters.
So the cell itself can modify the message.
Yes, directly influencing its local concentration and duration of action. For instance, certain cells can convert the less active thyroid hormone T4 into the much more potent T3 right there on site. This localized control adds another layer of specificity.
Okay. And what about the presence of other necessary intracellular factors?
Right. A hormone might bind to its receptor perfectly, signaling a command. But if the downstream cellular machinery, the signaling proteins, the enzymes, the cofactors, isn't present or functional, then the desired response simply won't occur.
Like having the key, but the door mechanism is broken.
Exactly. It's like a perfectly delivered message that can't be carried out because the necessary tools aren't available. The cell needs to be ready to receive and act on the signal.
That's a powerful analogy. We also have up or down regulation of the receptor due to ligand interaction. What does that mean exactly?
This refers to the cell's remarkable ability to adjust the number of receptors on its surface or within its interior in response to sustained hormone levels.
So it can change its sensitivity.
Precisely. If a cell is constantly bombarded by high hormone concentrations, it might downregulate its receptors, effectively reducing its sensitivity to prevent overstimulation. Conversely, if hormone levels are consistently low, the cell might upregulate its receptors, becoming more sensitive to capture even faint signals.
Like turning the volume up or down.
It's a built-in cellular thermostat, yes.
And finally, there's post-receptor desensitization of the cell. How is that different from downregulation?
This mechanism means that even if the hormone binds to its receptor and the receptor is present, the cell simply becomes less responsive to the signal after that binding event. This can involve changes in the signaling pathways downstream from the receptor.
So the signal gets dampened further down the line.
Exactly, acting as a brake on the hormonal response, even if binding is happening. These intricate factors demonstrate just how finely tuned and adaptable the target cell's response is, ensuring the body maintains a dynamic, healthy balance.
With that incredible overview of target cells, let's zero in on the true orchestrators of this specificity and action. Hormone receptors themselves. They're the unsung heroes of cellular communication.
Absolutely critical players. Hashtag AIPAW. Precision and discrimination of receptors. The challenge for these receptors is truly remarkable. Hormones circulate at incredibly low concentrations in the extracellular fluid. We're talking femto to nanomolar range. That's 10 to the minus 15 to 10 to the minus 9 moles per liter.
That sounds vanishingly small.
It is. To put that in perspective, this is like finding a single specific drop of dye perfectly placed in a swimming pool full of water. Meanwhile, your blood is teeming with other molecules, some structurally similar, at concentrations thousands to millions of times higher.
So the cell needs to pick out these tiny specific needles from an enormous chaotic haystack. This seems almost impossible for a biological system to manage.
It does, but that's precisely what these cell-associated recognition molecules, the receptors, accomplish. They provide an extraordinarily high degree of discrimination.
They're super filters.
They are. Receptors are exquisitely designed to distinguish between different hormones present in minute amounts, and critically, they can differentiate their specific hormone from a 106 to 109-fold excess of other similar or more abundant molecules.
Yeah.
It's an almost perfect molecular filter. Our source, HARPER's, illustrates this beautifully. A cell might have no receptors, or one type, or many types, and it must selectively bind only the relevant hormone, ignoring all the noise.
That's truly incredible. So how do we know if a hormone-receptor interaction is actually physiologically relevant? What are the gold standard criteria?
Yeah, good question. For an interaction to be considered physiologically relevant and truly defining a receptor, it must meet several biochemical features. First, the binding must be specific.
Meaning only the right key fits the lock.
Exactly. This means the hormone's binding to the receptor should be displaceable only by another molecule that mimics the hormone's action, an agonist, or one that blocks it, an antagonist. It's like a highly specific key fitting only one lock, and you can prove it's the right lock by trying a master key or a key designed to jam it.
A great analogy. What's next?
Second, the binding must be saturable. Receptors exist in a finite number on or within a cell. This implies there's a limited number of binding sites.
So you can fill them all up.
Right. Once all available receptors are occupied by the hormone, increasing the hormone concentration further will not lead to more binding. This demonstrates a finite capacity for recognition, which is a hallmark of a receptor, unlike a simple, non-specific binding event.
Makes sense. And the third criterion.
Third, the binding should occur within the concentration range of the expected biological response. If a hormone only binds to its receptor at unnaturally high, superphysiological concentrations levels never seen in the body, then that binding, while it might occur, isn't relevant to its normal function.
It has to work at normal body levels.
Exactly. The binding affinity should perfectly match the physiological concentration needed to elicit a response. Lastly, it's important to note that hormone-induced actions generally terminate when the hormone molecule, the effector, dissociates from the receptor. This ensures that the cellular response is transient and can be regulated.
So it can turn off again.
Right, preventing overstimulation and allowing the cell to adapt to changing signals. ##B, dual functional domains of receptors.
So these receptors aren't just passive binding sites that simply hold onto the hormone, are they? They have to actually do something after binding the hormone.
Exactly. They're not just docking stations. All functional receptors possess at least two critical functional domains, acting like two distinct parts of a sophisticated machine.
Okay.
The first is the recognition domain, which, as the name suggests, is solely responsible for binding the hormone ligand with that incredible high specificity and affinity we just discussed.
That's the listening part.
That's where the message is received, yes.
Yeah, and the second domain is where the real action happens, the signal generation part.
Precisely. That's the coupling domain. This domain links the hormone's recognition to an intracellular function, essentially initiating a signal within the cell. This process is known as signal transduction.
Translating the message.
It's the crucial step where the external message, carried by the hormone, is translated into an internal cellular action or command.
So this receptor-effector coupling is where the initial amplification begins, which is vital for those femtomolar concentrations to have a significant effect.
Yes, this coupling provides the very first step in the amplification of the hormonal response. A single hormone molecule binding to a single receptor can trigger a cascade of intracellular events, massively amplifying that initial faint signal.
That amplification is key.
Absolutely. This fundamental amplification is what differentiates a true receptor from a mere carrier protein.
And that leads us to a really important distinction, one that's often misunderstood. The difference between hormone receptors and those plasma carrier or transport proteins.
Yes, critical distinction.
They both bind hormones, but their roles are fundamentally different, as highlighted in our source.
This is a critical point for anyone aiming for that mastery level understanding. Let's compare them directly. For concentration, Receptors are present in very low numbers, typically just thousands per cell.
Tiny amounts.
Tiny. Plasma transport proteins, by contrast, are in high concentrations, billions per microliter of plasma. So many carriers, but very few highly specialized receptors.
What about their binding affinity? How tightly do they hold on?
Receptors exhibit high binding affinity, typically in the picomolar to nanomolar range. This ensures they can bind effectively to hormones even when they are present at extremely low physiological concentrations.
Beautiful.
Pretty much. Transport proteins, on the other hand, have lower binding affinity in the micromolar range. They bind a large quantity of hormone but less tightly.
Why less tightly?
Well, that's crucial because it allows the hormone to readily dissociate and become free and active when needed. It can't be locked up too tightly.
Okay. And their specificity?
Receptors possess very high specificity. They are designed to discriminate precisely between a specific hormone and countless other molecules, making them incredibly selective.
Only one key.
Right. Transport proteins have lower specificity. They might bind several structurally similar molecules, serving more as a general reservoir for a class of compounds.
Are they saturable? Can they get filled up?
Yes. Receptors are saturable because their number is limited. Once all binding sites are occupied, no more hormone can bind. Transport proteins, while they do have a finite capacity and can theoretically be saturated, their high concentration means they rarely reach saturation under normal physiological conditions for their primary cargo.
Okay. And reversibility. Can the hormone come off again?
Yes, both receptors and transport proteins exhibit reversibility in their binding. The hormone can bind and then dissociate, which is essential for dynamic regulation and the rapid termination of a response when the signal is no longer needed.
Finally, and most importantly, what about signal transduction? Passing the message along.
This is the defining difference that separates them. Receptors perform signal transduction. They translate hormone binding into an intracellular message that changes cellular behavior.
They do something with the message.
Exactly. Transport proteins, however, do not perform signal transduction. Their role is purely to carry and buffer the hormone. This distinction is paramount to understanding how hormones exert their profound and varied effects. ###C. Two general ways of signal transduction connecting to the cell's interior.
So how do these receptors actually generate that signal and get the message into the cell's interior? There are two general and fundamental ways this signal transduction occurs, depending on the type of hormone.
Right, two main strategies. The first category involves plasma membrane receptors. These are typically used by hormones that are water-soluble like polypeptide and protein hormones, and catecholamines.
The ones that can't just walk through the cell wall.
Exactly. Because these hormones cannot easily cross the cell's lipid membrane, they bind to receptors located on the plasma membrane. This binding then triggers a signal that propagates inside the cell, often by altering the activity of existing enzymes or other proteins.
And what are some of the key pathways and second messengers involved here, those internal signals?
Absolutely. Cyclic adenosine monophosphate, CMMP, is a classic and very widespread example. Many polypeptide hormones and catecholamines bind to their receptors on the cell surface, which then activate G proteins.
G proteins, okay.
These G proteins, typically characterized by 7 membrane-spanning domains, then switch on or off an enzyme called adenyl cyclase, which converts ATP into CAMP. This burst of CAMP then activates protein kinases.
Which are like little switches themselves.
Exactly. They phosphorylate other proteins, adding phosphate groups, leading to a cascade of effects within the cell. It's a major amplification pathway.
But we also hear about other pathways for plasma membrane receptors, like the JAK-STAT pathway, which sounds a bit different.
It is. For example, growth hormone, GH, and prolactin, PRL receptors, span the plasma membrane but uniquely lack their own intrinsic protein kinase activity.
So they don't phosphorylate things themselves.
Correct. Instead, when their ligand binds, it causes the receptor to recruit and activate cytoplasmic protein kinases known as Janus kinases, or JAKs. These activated JAKs then phosphorylate other proteins called STATs, which then translocate to the nucleus to regulate gene expression. So instead of directly activating enzymes inside the cell, this pathway acts like a rapid messenger service to the cell's nucleus, directly influencing which genes get turned on.
A direct line to the control center.
Pretty much, yeah.
And then there's CGMP, which often gets grouped with CalMPP, but it's distinct.
It is. Cyclic guanosine monophosphate, CGMP, serves as the second messenger for hormones like atrial natriuretic factor, ANF, and also for nitric oxide. These pathways often lead to smooth muscle relaxation and are critical in regulating processes like blood pressure.
Many hormones are linked to calcium or phosphoinositides or both. That seems like a very busy dynamic pathway.
Indeed. Rapid changes in ionic calcium, C2+ levels, or the metabolism of phosphoinositides are crucial intracellular second messengers for a broad range of hormones.
Like which ones?
This includes acetylcholine, I1 adrenergic catecholamines, angiotensin II antidiuretic hormone, calcitriol, gastrin, gonadotropin-releasing hormone, oxytocin, platelet-derived growth factor, PDGF, substance P, and thyrotropin-releasing hormone, TRH. Phew.
That's a long list.
It is. These pathways often involve rapid changes in intracellular calcium levels or the generation of lipid-derived messengers, leading to a variety of swift and potent cellular responses.
And finally, the vast category of kinase or phosphatase cascades. This sounds like a relay race of phosphorylation.
That's an excellent way to describe it. A wide variety of hormones utilize complex kinase or phosphatase cascades as their second messengers. Here, one enzyme phosphorylates or dephosphorylates another, which then modifies a third, creating a chain reaction that amplifies the signal and leads to specific cellular outcomes. Examples include adiponectin, epidermal growth factor, EGF, erythropoietin, EPO, fibroblast growth factor, FGF, growth hormone, GH, insulin, insulin-like growth factors, leptin, and prolactin. And importantly, a given hormone may, in fact, use multiple kinase cascades to elicit its full range of effects.
Wow, layers upon layers.
Highlighting the intricate web of intracellular signaling and the cell's sophisticated internal language.
So that covers the surface receptors. What about hormones that just walk right into the cell? The fat-soluble ones.
That brings us to the second general way of signal transduction, intracellular receptors. These are utilized by lipophilic hormones like steroid hormones, retinoids, and thyroid hormones.
They can slip through the membrane.
Exactly. Because of their lipid-soluble nature, these hormones can readily traverse the plasma membrane and interact with the receptors located inside the cell, either in the cytoplasm or directly within the nucleus.
And here, the hormone receptor complex itself is the signal, directly affecting gene transcription. That's a very direct form of communication.
Precisely. Once the hormone binds to its intracellular receptor, the resulting ligand receptor complex directly provides the signal. This complex then typically binds to specific DNA sequences, generally affecting the rate at which certain genes are transcribed into RNA.
Controlling which proteins get made.
Ultimately, yes, controlling which proteins are made. This is a slower but often more sustained form of hormonal action compared to the rapid enzyme modifications seen with membrane receptors.
And these receptors are part of a larger nuclear receptor superfamily, right?
Yes. Scientists discovered that steroid receptors shared remarkable similarities in their amino acid sequences with thyroid hormone receptors, especially in their DNA binding regions. This led to the realization that these receptors are members of a large nuclear receptor superfamily.
And they're orphans in this family.
That's the fascinating part. Many related members of this family currently have no known ligand, leading them to be termed orphan receptors, hinting at undiscovered hormones or signaling pathways. This superfamily plays a critical role in how hormones directly regulate gene expression, essentially turning genes on or off.
And they must have distinct structural domains to perform all these functions, binding hormone, binding DNA, talking to other proteins.
They do. These receptors possess several functional domains, each like a specialized compartment. One specific site that binds the hormone, another domain that binds to specific DNA sequences, often called hormone response elements, a third region involved in interacting with various co-regulator proteins, which can either amplify or repress gene transcription, and sometimes a fourth region that influences how the receptor moves within the cell.
Modular design.
Very much so. This modular design allows them to precisely regulate gene expression in a highly coordinated fashion. #tag-tag-tag-D, specific examples of receptor proteins.
To make this concrete, can you give us a specific example of one of these receptor proteins? The insulin receptor is a classic.
The insulin receptor is an excellent example of a complex plasma membrane receptor. It's a heterotetramer, meaning it's composed of two copies of two different protein subunits, specifically two alpha and two beta subunits, all linked together by multiple disulfide bonds.
So 4 parts linked together. And how do these subunits work together to relay the signal?
The extracellular alpha subunit is entirely responsible for binding insulin. It sits outside the cell waiting. Once insulin binds, the signal is transduced across the membrane through the beta subunit, which spans the cell membrane.
So the beta part goes through the wall.
Right. And crucially, the cytoplasmic portion of the beta subunit, the part inside the cell, contains an intrinsic tyrosine protein kinase domain.
Meaning it's an enzyme itself.
Exactly. The receptor itself is an enzyme that, upon insulin binding, phosphorylates or adds phosphate groups to specific tyrosine residues on other proteins inside the cell. This initiates the signaling cascade that leads to insulin's profound metabolic effects, such as glucose uptake.
And other receptors work like this too.
Yes, the receptors for insulin-like growth factor, IGFI, and epidermal growth factor, EGF, are structurally quite similar, sharing this elegant tyrosine kinase mechanism, highlighting a common efficient design for cellular communication.
Now that we have a solid understanding of how hormones act and how their receptors work, let's zoom out to the bigger picture. How we classify these incredibly diverse chemical messengers. It helps us organize this complex system.
It does, yeah. There are a few ways to slice it. #tag John classification criteria. Hormones can be classified in several different ways, which provides us with multiple lenses to understand their varied nature. We can classify them according to their chemical composition, what they're fundamentally made of, like steroids or proteins.
Okay, what they're built from.
We can also categorize them by their solubility properties, whether they're water-soluble or fat-soluble, which dictates how they travel.
How they handle water.
Right. The location of their receptors, whether on the cell surface or inside the cell, is another key criterion, defining how they interact with the cell. And finally, we classify them by the nature of the intracellular signal they use to mediate their action, referring to the second messengers involved.
So, how they talk inside the cell.
Exactly. For our deep dive, classifying them by receptor location and their intracellular signaling mechanism is particularly insightful for understanding their functional differences. It groups them by how they work. #B, group I hormones, lipophilic intracellular receptors.
Let's start with group I hormones. These are the ones that are generally lipophilic, meaning fat-loving, and they use intracellular receptors.
Correct. The characteristics of group I hormones are quite distinct. They include all the steroid hormones like cortisol and testosterone, iodothyronines, which are your thyroid hormones T3 and T4, calcitriol, the active form of vitamin D, and retinoids, like retinoic acid.
All the fatty ones.
Basically, yes. Because they are lipophilic, they simply wouldn't dissolve well in your watery blood plasma.
So they need transport proteins because they wouldn't dissolve well in plasma otherwise. It's like needing a special boat to cross a lake if you can't swim.
Exactly. After secretion from their producing gland, these hormones associate with specific plasma transport or carrier proteins. This serves a dual purpose. It solves their solubility problem, allowing them to be delivered efficiently throughout the body, and it also significantly prolongs their plasma half-life.
They last longer in the blood.
Right, often ranging from hours to days because they're protected from rapid degradation when bound to these carriers.
And only the free, unbound form is biologically active, right? The stuff not on the boat.
Yes, that's a fundamental principle. Only the free unbound hormone is biologically active and can readily diffuse across the cell's lipid plasma membrane. Once inside the target cell, they find their receptors either in the cytoplasm or directly within the nucleus.
And then what happens?
For group I hormones, the hormone receptor complex itself acts as the intracellular messenger, directly influencing gene expression by binding to specific DNA sequences. It's a direct command from the hormone to the cell's genetic machinery.
Straight to the DNA control panel. Can you give some key examples of these group I hormones?
Certainly. This group includes androgens, like testosterone, driving male characteristics, calcitriol, regulating calcium, estrogens, key for female reproductive health, glucocorticoids, like cortisol, involved in stress response, mineralocorticoids, like aldosterone, regulating salt and water, progestins, like progesterone, important in pregnancy, retinoic acid, involved in growth and development, and the thyroid hormones, T3 and T4, regulating metabolism. A really important group, #C, group 2 hormones, water-soluble cell surface receptors.
In contrast, we have group 2 hormones, which are generally water-soluble and bind to cell surface receptors. This means they need a different strategy.
Completely different. These hormones, which encompass polypeptides, proteins, glycoproteins, and catecholamines, are hydrophilic. They love water.
Happy in the blood.
Exactly. Consequently, they are freely soluble in plasma and generally do not require transport proteins. This also means they are more susceptible to enzymatic degradation in the bloodstream, so they typically have a short plasma half-life, usually measured in minutes.
Fast acting, fast disappearing.
Generally, yes. Their actions are often rapid and transient.
And because they can't cross the membrane, they're receptors on the plasma membrane, so they need a way to get the message inside without entering the cell themselves.
Precisely. They bind to specific receptors spanning the plasma membrane of the target cell. To communicate with intracellular metabolic processes, they rely on intermediary molecules called second messengers.
The internal relay team.
Right. The hormone itself is considered the first messenger. This concept arose from the observation that epinephrine, for example, binds to the plasma membrane and increases intracellular CAMP, which was then found to mediate the effects of many other hormones. It's like someone ringing your doorbell, the hormone, and you sending out a different person, the second messenger, to relay the message inside.
Okay, so what are the different subcategories of group 2 hormones based on their second messengers? How do they relay that message?
We can broadly categorize them based on the major second messenger systems they activate. One large group uses CAM as a second messenger.
That's a classic one.
It is. Think of hormones like adrenaline acting via obrinergic receptors, ACTH, vasopressin, glucagon, and parathyroid hormone. These often lead to rapid changes in cellular metabolism or secretion.
Okay.
Another group employs CGMP as a second messenger. Key examples here are atrial natriuretic factor, ANF, which helps regulate blood pressure, and nitric oxide, a crucial local signaling molecule.
Right.
A very important and diverse group utilizes ionic calcium, calcium CN2+, or phosphoinositides, or both, as their second messenger.
A calcium signal.
Yes, this includes hormones like acetylcholine, acting via muscarinic receptors, angiotensin II, oxytocin, and thyrotropin-releasing hormone, TRH. These pathways often involve rapid increases in intracellular calcium, leading to muscle contraction, secretion, or other quick responses.
And the last group, cascades.
And finally, a large category transduces signals through kinase or phosphatase cascades. These involve chains of enzymes that phosphorylate or dephosphorylate each other, amplifying and diversifying the signal. Key hormones here are insulin, growth hormone GH, epidermal growth factor EGF, and erythropoietin, EPO.
A lot of growth factors in there.
Yes, and it's important to remember that a single hormone might even activate multiple kinase cascades or utilize slightly different second messengers depending on the specific target cell, highlighting the amazing adaptability of these systems.
Now that we've broadly classified hormones and understood how they act, let's really dive into the incredible diversity of the endocrine system itself, from how hormones are synthesized to how they're stored. This is where we truly appreciate the body's biochemical ingenuity.
Yeah, the variety is stunning. #tag #A. Diverse cellular arrangements for hormone synthesis. The body has evolved a fascinating array of cellular arrangements for hormone synthesis, each precisely tailored to specific needs and regulatory demands. We see everything from organs solely dedicated to hormone production to specialized cells tucked away within other organs, and even remarkable multi-organ collaborations.
So for organs dedicated to hormone production, we're talking about structures like the thyroid gland that primarily produces thyroid hormones, or the adrenal glands that churn out stress hormones like cortisol and mineralocorticoids.
Right. And the pituitary gland, the master gland, also produces a whole suite of hormones like TSH, FSH, LH, GH, PRL, and ACTH. Their main job is hormone creation and secretion.
Then we have organs designed for dual functions, performing two distinct but closely related roles.
Exactly. The ovaries, for instance, don't just produce mature oocytes for reproduction. They're also responsible for the crucial reproductive hormones estradiol and progesterone. Similarly, the testes produce mature spermatozoa alongside testosterone.
Functionally linked.
Tightly intertwined, yes. In these cases, the reproductive function and hormonal function are closely linked.
And beyond entire organs, some hormones are made in specialized cells within other organs. This is quite clever, allowing for localized control.
Indeed. Consider the small intestine, which contains specialized cells that produce glucagon-like peptide, important for glucose regulation. The thyroid gland, beyond T3 and T4, also has specific cells for calcitonin, which helps regulate calcium.
Okay.
And the kidney, a vital organ for excretion and filtration, also produces angiotensin II, a crucial hormone in blood pressure regulation. These cells are embedded within larger organs, contributing to broader physiological functions.
And the most complex arrangement must be when multiple organs work together to synthesize a single hormone. That sounds like a true biochemical relay race.
That's precisely the case with 1025-OHTT3 or calcitriol, the active form of vitamin D. Its production requires a coordinated effort involving cells in the skin, the liver, and the kidney.
Skin, liver, kidney stuff.
Showcasing a remarkable inter-organ collaboration. This diversity in the approach to hormone synthesis illustrates the evolutionary fine-tuning to fulfill specific physiological purposes, ensuring that hormones are made where and when they are needed. #B, chemical diversity of hormones.
It's not just the locations that are diverse.
Yeah.
The hormones themselves are built from a wide variety of chemical building blocks, demonstrating incredible molecular craftsmanship.
Yes, the chemical diversity is striking. A large series of hormones, for instance, are all derived from cholesterol.
Cholesterol gets a bad rap, but it's essential here.
Absolutely essential. This includes the glucocorticoids, like cortisol, mineralocorticoids, like aldosterone, androgens, like testosterone, estrogens, like estradiol, progestins, like progesterone, and 1025-OH2D3. What's truly remarkable here is how a single starting molecule cholesterol can be sculpted into dramatically different hormones by just a handful of specialized enzymes in specific cells.
Molecular sculpture.
It's a masterclass in biochemical efficiency and specificity.
And what's interesting is that sometimes one steroid hormone is actually a precursor for another. It's like a family tree of hormones.
Absolutely. Progesterone, while a hormone in its own right, also serves as a crucial precursor for the formation of glucocorticoids, mineralocorticoids, testosterone, and estrogens.
Wow, very central.
Likewise, testosterone is an obligatory intermediate in the biosynthesis of estradiol and in the formation of dihydrotestosterone, DHT. In these cases, the final hormone produced is determined by the specific cell type and the unique set of enzymes present within that cell. This highlights the exquisite cellular specificity that dictates the hormonal landscape of different tissues.
Let's really drill down on this. How are adrenal steroid hormones, like cortisol and aldosterone, synthesized from cholesterol? This sounds incredibly precise.
It is, and it's a testament to biochemical precision. Adrenal steroid hormones are primarily synthesized from cholesterol, most of which comes from the plasma. A smaller portion can be synthesized from scratch within the adrenal gland itself.
Okay.
Much of this cholesterol is stored in cytoplasmic lipid droplets within the adrenal cells.
So it's stored, but then what triggers its use to make hormones, like stress?
Exactly. Upon stimulation of the adrenal gland by ACTH, a key pituitary hormone often released during stress, an enzyme is activated that releases free cholesterol from these droplets. This free cholesterol is then transported into the mitochondrion, the cell's power factory.
Into the powerhouse.
Right. Here, a critical step occurs. The transport of cholesterol into the mitochondrion by a special protein called steroidogenic acute regulatory STAR protein. STAR is essential for getting cholesterol to the inner mitochondrial membrane where the first enzymatic conversion takes place.
And that's the slow step.
This is often the rate-limiting step for steroid hormone production, controlling the delivery of the raw material.
And what's that initial conversion once cholesterol gets into the mitochondria?
Inside the mitochondrion, a specialized enzyme system converts cholesterol into pregnenolone. Think of this enzyme as a molecular scissor, clipping off a specific part of the cholesterol molecule to yield a smaller 21-carbon steroid. Pregnenolone is then the universal starting point for all mammalian steroid hormones.
The common ancestor molecule.
Yes.
Pregnenolone is the common ancestor. What about the enzymes involved and where they're located?
All steroid hormones are formed from pregnenolone through a series of reactions that occur in either the mitochondria or the endoplasmic reticulum of the producing cell. These reactions often involve enzymes that add oxygen atoms, hydroxylases, or remove hydrogen atoms, dehydrogenases, or reshape the molecule, isomerases and lyases.
Different tools for different jobs.
Exactly. And what's critical is the cellular specificity. For example, the unique enzymes required for aldosterone synthesis are found only in the outermost region of the adrenal cortex, the zona glomerulosa. This means aldosterone production is strictly confined to that specific area.
Let's focus on the mineralocorticoid synthesis pathway, which produces aldosterone, crucial for salt and water balance. How does that work in the zona glomerulosa?
Aldosterone synthesis follows a specific pathway, occurring exclusively in the zona glomerulosa. The journey from pregnenolone involves a few key transformations. It's converted to progesterone, which then gets modified by enzymes that add oxygen atoms at specific points. One key enzyme adds an oxygen at carbon 21, forming 11-deoxycorticosterone, or DOC,
EOC.
which itself has significant salt-retaining activity. Then another enzyme adds an oxygen at carbon 11, leading to corticosterone. Corticosterone does have some glucocorticoid and weak mineralocorticoid activity, and in some species it's actually the main stress hormone.
And what's unique to the zona glomerulosa for aldosterone? What makes it special there?
The zona glomerulosa lacks certain enzymes needed for other steroids, but it uniquely possesses a mitochondrial enzyme often called aldosterone synthase. This enzyme is a master sculptor that takes corticosterone and, through a series of precise modifications, including adding an oxygen at carbon 18 and then converting that into an aldehyde, it yields aldosterone.
A very specific final step.
Extremely specific. This very specific enzyme distribution, along with the tight regulation of the zona glomerulosa by potassium levels and a hormone called angiotensin II, is so distinctive that some researchers view this adrenal region as almost a separate mini organ in its function.
That's a great point of detail. Now, for glucocorticoid synthesis, focusing on cortisol, our primary stress hormone, where does that happen?
Cortisol synthesis occurs primarily in the middle and inner zones of the adrenal cortex, the fasciculata and reticularis. This pathway also starts from pregnenolone but takes a different enzymatic route, involving specific additions of oxygen at carbon 17, then carbon 21, and finally carbon 11.
And the order matters.
Yes, the order is crucial. The C11 oxygen addition is often the slowest step, acting as a bottleneck for overall cortisol production.
What happens if the C11 oxygen is added first?
If the C11 oxygen is added too early in the sequence, it actually diverts the pathway towards mineralocorticoid production instead of glucocorticoids. This highlights the precise order of operations required.
Fascinating.
The enzyme that adds oxygen at carbon 17 is a key player, acting on pregnenolone or progesterone. From a 17 oxygenated intermediate, another enzyme adds an oxygen at carbon 21, and finally, a mitochondrial enzyme adds the last crucial oxygen at carbon 11, resulting in cortisol, the most potent natural human glucocorticoid.
And this involves moving things around in the cell.
Yes, this pathway involves substrates repeatedly moving between the mitochondria and the endoplasmic reticulum, showcasing the dynamic cellular environment of steroidogenesis.
Moving on to adrenal androgen synthesis, which produces precursors for sex hormones. What's the main one there?
The primary androgen, or androgen precursor, produced by the adrenal cortex is dehydroepiandrosterone, or DHEA. A small portion of an intermediate in the cortisol pathway gets diverted by a specific enzyme that clips off part of its side chain.
And that enzyme is P450C17, right? The multi-tool enzyme.
Correct. This enzyme, P450C17, is fascinating because it has a dual function. It can add an oxygen at carbon 17, and it also has this clipping activity, the 17,020 lyase activity. So it's like a multi-tool enzyme.
And DHEA itself isn't that strong.
Right. DHEA itself is considered a prohormone. It's relatively weak, but it can be converted into more potent androgens like androstenedione and even testosterone in other tissues, including the gonads.
So the adrenals make the building blocks.
Exactly. The adrenal gland provides the building blocks, and other tissues do the final construction. Clinically, if there's a problem with cortisol synthesis, the adrenal gland might overproduce these androgen precursors, leading to conditions like adrenogenital syndrome, where androgen levels are abnormally high.
So speaking of the testes, let's turn to testicular steroidogenesis and the biosynthesis of testosterone in the Leydig cells. How does that compare to adrenal synthesis?
The immediate precursor of gonadal steroids, like adrenal steroids, is cholesterol. And similar to the adrenal gland, the rate-limiting step in testosterone biosynthesis is getting cholesterol into the inner mitochondrial membrane, again mediated by that crucial transport protein, STAR.
It's our protein again.
Once cholesterol is in the correct mitochondrial location, it is converted to pregnenolone by the same molecular scissor enzyme, P450CCE, we saw in the adrenal. However, in the testes, this initial step is primarily stimulated by luteinizing hormone LH, not ACTH.
Different trigger. And what's the enzymatic path from pregnenolone to testosterone? Still using that multi-tool enzyme.
Yes. The conversion of pregnenolone to testosterone requires a series of 5 distinct enzyme activities that are carried out by 3 different proteins. These include enzymes that add oxygens and reshape the molecule, and crucially, the same P450C17 enzyme that we saw in the adrenal gland, which again acts as a dual function protein, adding oxygen at carbon 17 and performing that clipping activity.
So there are 2 main pathways, right? A progesterone pathway and a DHEA pathway. Which one is more common?
That's right. The pathway involving progesterone proceeds step-by-step to testosterone. Alternatively, pregnenolone can be converted to testosterone via the DHEA pathway. In humans, this DHEA route actually appears to be the predominant pathway used in the testes.
Interesting.
These enzyme activities are precisely localized within specific parts of the cell, allowing for efficient and regulated testosterone production.
Now, a key point for testosterone's activity is its conversion to dihydrotestosterone, or DHT, in peripheral tissues. How does that happen, and why is it so significant?
Testosterone is metabolized via two main pathways. One pathway leads to generally less active forms, but the second and biologically crucial pathway converts testosterone into DHT. This occurs primarily in specific target tissues.
So DHT is the real powerhouse in many tissues. Even if testosterone is the main circulating androgen from the testes, it gets supercharged locally.
Exactly. DHT is considered the most significant active metabolic product of testosterone because in many key target tissues, including the prostate, external genitalia, and certain areas of the skin, DHT, not testosterone, is the most potent active form of the hormone.
And is most DHT made in the testes or elsewhere?
While the testes secrete some DHT, the vast majority of DHT is actually produced peripherally from testosterone in the very cells where it needs to exert its most powerful effects.
And what enzyme is responsible for that conversion?
This crucial conversion is catalyzed by an enzyme called fiber reductase. This makes testosterone effectively a prohormone. It's produced by the testes, but then converted into a much more potent compound, DHT, largely outside the testes, closer to where the action is needed.
A very clever system.
It's also worth noting that some estradiol, an estrogen, is also formed from the peripheral aromatization of testosterone, especially in males, highlighting the interconnectedness of steroid metabolism.
Let's turn our attention to ovarian steroidogenesis and the production of estrogens. What's the main one?
The estrogens are actually a family of hormones synthesized in various tissues. 17-estradiol is considered the primary estrogen of ovarian origin, playing a critical role in female reproductive health. The early steps of estradiol synthesis are quite similar to those involved in androgen biosynthesis, but there are unique features specific to the ovary.
So how are estrogens formed from androgens? It sounds like another conversion, like DHT from testosterone.
It is another conversion. Estrogens are formed by the aromatization of androgens. This is a complex biochemical process that involves several specific steps, requiring oxygen and reducing power from NADPH. The key enzyme complex here is called aromatase.
Aromatase.
If the substrate for this enzyme complex is testosterone, the product is estradiol. If the substrate is androstenedione, the result is estrone. Aromatase essentially takes an androgen and transforms its structure to create an estrogen.
And there's a two-cell model that explains how different ovarian cells contribute to this process, right? A collaboration?
Yes, the cellular source of various ovarian steroids involves a fascinating transfer of substrates between two cell types, often referred to as the two-cell model. The Thuca cells are the primary ovarian source of androstenedione and testosterone.
That makes the androgens.
Right. These androgens are then transferred to the adjacent granulosa cells. The granulosa cells contain the aromatase enzyme, which then converts these androgens into estrone and estradiol, respectively.
So the granulosa cells do the aromatization step.
Exactly. It's a beautiful example of cellular cooperation.
It's not just ovarian cells making estrogens, though. Peripheral aromatization is crucial for overall estrogen levels, isn't it? Especially outside the reproductive years.
That's a critical point. Significant amounts of estrogens are also produced by the peripheral aromatization of androgens outside the ovaries. For example, in human males, the peripheral aromatization of testosterone to estradiol, E2, accounts for as much as 80% of the latter's production.
Wow, 80% in males.
Yes. In females, adrenal androgens serve as important substrates, with a substantial portion of E2 produced during pregnancy coming from the aromatization of these adrenal androgens. Furthermore, the conversion of androstenedione to estrone is the major source of estrogens in postmenopausal women as ovarian estrogen production declines.
And this peripheral aromatase activity has clinical implications for things like obesity or certain cancers.
Absolutely. Aromatase activity is not just in the gonads. It's present in adipose, fat cells, liver, skin, and other tissues. Increased activity of this enzyme can contribute to certain conditions, such as estrogenization, seen in liver cirrhosis, hyperthyroidism, aging, and obesity. This understanding has led to important therapeutic advancements. Aromatase inhibitors are proving very promising as therapeutic agents in breast cancer and potentially other female reproductive tract malignancies by blocking estrogen synthesis in these peripheral tissues.
Let's shift to another cholesterol derivative.
Yeah.
1025-OH2D3, or calcitriol, which plays a vital role in calcium homeostasis. Its synthesis is incredibly complex, involving multiple tissues working in sequence, the relay race we mentioned earlier.
Indeed, 1025-OH2D3 is produced by a complex series of enzymatic reactions that necessitate the transport of precursor molecules through the plasma to several different tissues. It's often mistakenly called vitamin D, but it's not a true vitamin because your body can synthesize it endogenously.
So we make our own vitamin D.
We do. The active molecule, 1025-OH2D3, is then transported to other organs where it activates biological processes in a manner similar to that employed by steroid hormones.
And it all starts in the skin, which is surprising for a hormone that's also considered a vitamin, triggered by sunlight.
It does. While small amounts of its precursor are found in foods, most of the precursor for 1025-OH2D3 synthesis is produced right there in your skin. Specifically, a cholesterol derivative called 7-dehydrocholesterol undergoes a non-enzymatic reaction triggered by ultraviolet UV light to form pre-vitamin D3.
And more sun means more production.
Generally, yes. The efficiency of this conversion depends directly on the intensity of UV exposure and inversely on the extent of skin pigmentation. Interestingly, as we age, there's a natural loss of 7-dehydrocholesterol in the skin, which may contribute to calcium balance issues often seen in older individuals.
So skin produces a precursor, then it goes to the liver for the next step in the relay.
That's correct. A specific transport protein called the vitamin D binding protein carries vitamin D3 from the skin or intestine to the liver. In the liver, it undergoes its first essential modification, a hydroxylation, adding an O2H group at position 25.
Okay, step 1 in the liver. Is that step regulated?
Interestingly, no. This reaction takes place in the endoplasmic reticulum and involves specific enzymes, but this particular reaction in the liver is not tightly regulated. It mostly just happens. The product, 25-OH2D3, becomes the major form of vitamin D circulating in your plasma, and is then transported to the kidney by the same vitamin D binding protein, ready for the final activation.
And finally, the kidney is where it gets its full power, the last leg of the race.
Yes. 25-OH2D3 is only a weak signal on its own. To achieve full biological activity, it must be further modified by hydroxylation at position 1. This crucial activation step takes place in the mitochondria of the renal proximal convoluted tubules in the kidney.
Another mitochondrial step.
Yes. It's a sophisticated enzymatic process involving a cytochrome P450 system that produces 1025-OH2D3, which is the most potent naturally occurring metabolite of vitamin D. It's now fully active and ready to exert its profound effects on calcium and phosphate homeostasis throughout the body.
Beyond cholesterol, another critical building block for hormones is the amino acid tyrosine. This humble amino acid gives rise to two very different, yet vitally important, classes of hormones, catecholamines and thyroid hormones.
That's right. Both are synthesized from tyrosine, but their structures, synthesis pathways, and functions are remarkably distinct, showcasing the versatility of biological chemistry.
Let's start with catecholamines. Dopamine, norepinephrine, and epinephrine. We know them for their roles in fight or flight. Where are they made and how are they handled?
These three amines are primarily synthesized from tyrosine in specialized cells called chromaffin cells within the adrenal medulla, which is the inner part of your adrenal glands.
The core of the adrenal gland.
Exactly. Epinephrine, often called adrenaline, is the major product of the adrenal medulla, constituting about 80% of its catecholamines, and it's uniquely not made in other tissues. Norepinephrine, on the other hand, is mostly made directly where it's needed, in the sympathetic nerves that connect to various organs.
So they're synthesized in their final active form and then stored, waiting for release, like little grenades ready to go.
You could think of it that way. Correct. They are synthesized in their active form and then stored in specialized secretion granules within those chromaffin cells. Upon appropriate neural stimulation, say, during stress or excitement, these granules are released from the cell through exocytosis, discharging the catecholamines directly into the circulation. These cells maintain a significant reserve supply, enough for several hours of intense demand.
Wow, hours worth. What are the key sequential steps involved in their biosynthesis, starting from tyrosine?
The conversion of tyrosine to epinephrine requires 4 sequential enzymatic steps. The first step is a ring hydroxylation, where tyrosine is converted to L-DOPA. This reaction is catalyzed by tyrosine hydroxylase.
And that's the important control step.
Yes, which is the rate-limiting enzyme in catecholamine biosynthesis. This enzyme is highly regulated, notably by feedback inhibition from the catecholamines themselves, ensuring that production matches demand.
And there's a clinical connection to this rate-limiting step, isn't there? With Parkinson's.
Yes. Catecholamines cannot easily cross the blood-brain barrier. Therefore, in the brain, they must be synthesized locally. In certain central nervous system diseases, like Parkinson's disease, there's a deficiency of dopamine synthesis in specific brain regions. Since L-DOPA, the direct precursor of dopamine, readily does cross the blood-brain barrier.
The precursor gets through.
Exactly. It's used as an important therapeutic agent to boost dopamine levels in the brain to treat Parkinson's symptoms.
What's the second step after L-DOPA?
The second step is decarboxylation, removing a carboxyl group, where L-DOPA is converted to dopamine by DOPA decarboxylase. This enzyme is present in many tissues. Interestingly, compounds that resemble L-DOPA can act as competitive inhibitors of this reaction and are sometimes used to treat certain types of hypertension.
Okay. And then dopamine is converted to norepinephrine. Where does that happen?
That's the third step, side chain hydroxylation. Dopamine is converted to norepinephrine by an enzyme called dopamine hydroxylase, DBH. This conversion actually occurs within the secretion granules themselves.
Inside the storage vesicle.
Yes, meaning dopamine enters the granule and then norepinephrine is formed inside before release.
Clever. And the final step, getting to epinephrine, the main adrenal one.
The fourth step is N-methylation, adding a methyl group where norepinephrine is converted to epinephrine by an enzyme called phenylethanolamine N-methyltransferase, PNMT. This specific conversion happens in the cytoplasm of the epinephrine-forming cells of the adrenal medulla.
And PNMT is regulated by cortisol.
Yes, what's fascinating is that the synthesis of PNMT is strongly induced by glucocorticoid hormones like cortisol, which reach the adrenal medulla via a special intraadrenal portal circulatory system. This ensures that sufficient PNMT is available to produce the large amounts of epinephrine needed during stress, a direct link between stress hormones.
The thyroid hormones, T3 and T4, are also tyrosine derivatives, but their synthesis is uniquely complex, requiring a trace element that's often scarce globally: iodine.
Indeed. The formation of triiodothyronine, T3, and tetrodotyronine, thyroxine, T4, highlights many unique aspects of hormonal diversity. These hormones are unique because they absolutely require iodine for their biological activity.
No iodine, no thyroid hormone.
Essentially, yes. Given that dietary iodine is scarce in many parts of the world, an intricate mechanism for accumulating and retaining iodide has evolved within the thyroid gland. Furthermore, they are synthesized as part of a very large precursor molecule called thyroglobulin.
A giant protein scaffold.
Exactly. They're stored in a massive extracellular reservoir, and the less active T4 is peripherally converted to T3, which is the much more active hormone. In essence, T4 can be considered a prohormone with significant implications for how we understand its function.
So thyroglobulin is the central player here. What exactly is this precursor? How big is it?
Thyroglobulin, TGB, is the precursor for both T4 and T3. It's an exceptionally large iodinated and glycosylated protein made up of two large subunits, molecular weight around 660 kilodalton weight. Crucially, it contains many tyrosine residues, about 115, each a potential site for iodine attachment.
Lots of spots for iodine.
Lots of spots, but not all are used equally. About 70% of the iodine in thyroglobulin exists in inactive forms, mono and diiotyrosine, MIT and DIT, while only 30% is incorporated into the active T4 and T3 molecules. Thyroglobulin, with its massive size, provides the specific three-dimensional shape required for the proper coupling of tyrosine residues and the incorporation of iodine necessary for the formation of the thyroid hormones.
And where is it stored and how is it released? You said a huge reservoir.
Thyroglobulin is synthesized within the thyroid follicular cells and then secreted into the lumen of the thyroid follicle, where it is stored as a gel-like substance called colloid. This colloid actually represents a several weeks supply of T3 and T4 in a normal thyroid gland.
Weeks. That's a lot of backup.
It is, acting as a critical buffer. When the thyroid gland is stimulated by thyroid-stimulating hormone, TSH, the colloid is reabsorbed into the cells where it's broken down by enzymes, proteases, and peptidases. This hydrolysis releases T4 and T3, which are then discharged into the bloodstream. Thyroglobulin is truly a very large prohormone, serving as both factory and warehouse for thyroid hormone production.
Now let's break down iodine metabolism itself. How does the thyroid acquire and process this crucial element, step by step?
Iodide metabolism involves several discrete and highly regulated steps. First is concentration. The thyroid gland has an extraordinary ability to actively transport and concentrate iodide from your blood against a strong electrochemical gradient.
It sucks it in.
It does, using an energy-dependent process facilitated by a specific Na plus isoporter, or iodide transporter. The ratio of iodide in the thyroid to iodide in the serum can be remarkably high, like 25.1 normally, ensuring the gland can capture scarce iodine resources.
Okay, step 1, trap the iodine. What's next?
The second step is oxidation. The thyroid is unique in its ability to oxidize iodide to a higher reactive state, like I plus or I0, which is absolutely necessary for iodide to be incorporated into organic molecules, specifically the tyrosine residues on thyroglobulin.
And what enzyme does that?
This step involves a heme-containing enzyme called thyroperoxidase, TPO. Clinically, certain drugs, like Thury is used to treat an overactive thyroid, directly inhibit this iodide oxidation step.
Got it. Oxidation, then coupling.
The third step is coupling. This is where two modified tyrosine molecules, specifically two DITs within the thyroglobulin protein, are coupled together to form T4, or a single modified tyrosine MIT is coupled with another DOT to form T3.
So pairing them up.
Yes, pairing them up, right on the thyroglobulin backbone. This entire process occurs within the thyroglobulin molecule itself, and the same thyroperoxidase enzyme is thought to catalyze this crucial coupling step. The formed thyroid hormones remain integral parts of thyroglobulin until the molecule is degraded and they are released.
Okay, trap, oxidize, couple. What's the final step? Deiodination. Isn't that taking iodine off?
Yes, but it's important for recycling and activation. Within the thyroid gland itself, an enzyme called deiodinase removes iodine from inactive intermediates, MIT and DIT, effectively recycling iodide for further T3 and T4 biosynthesis, a vital conservation mechanism.
Smart recycling.
Very. More broadly, and perhaps more importantly for activity, a peripheral deiodinase enzyme found in target tissues like the pituitary, kidney, and liver selectively removes iodine from the 5' position of T4 to produce T3.
And activating T4 into the more potent T3.
Precisely. T3 is the much more biologically active molecule. In this sense, T4 can truly be thought of as a pro-hormone, awaiting activation in the tissues where it's needed most, even though T4 does have some intrinsic activity itself.
That's incredibly detailed. So many hormones like insulin and PTH and thyroid hormones are made from these larger peptide precursors. What's the general principle behind this multi-step processing? Why not just make the final product directly?
That's a great question. The general principle here is multifaceted, combining efficiency with precise control. In some cases, like insulin, the larger precursor molecule called proinsulin is crucial for achieving the correct three-dimensional structure and facilitating proper disulfide bond formation, which is essential for the mature hormone's activity.
So it helps it fold correctly.
Exactly. It's like using a scaffolding to build a complex structure and then removing the scaffolding once the structure is stable.
And for others, it's about control over active hormone availability, making sure you don't release too much too soon.
Exactly. For many other hormones, like parathyroid hormone, PTH, and angiotensin II, synthesizing them as a larger precursor serves as a sophisticated mechanism for tightly controlling the available amount of the active hormone. This allows for rapid post-translational regulation, modifying the protein after it's made, and quick adjustments of active hormone levels without needing to synthesize the entire protein from scratch every time.
Faster response time.
Potentially, yes. Another fascinating example is the pro-opiomelanocortin, POMC, protein, which can be processed into many different biologically active hormones in a tissue-specific manner, offering incredible regulatory flexibility from a single gene.
Let's focus on insulin, a crucial peptide hormone regulating blood glucose. How does its pre-prohormone processing work? You mentioned folding.
Insulin has a unique structure, an AB heterodimer, meaning it consists of two distinct protein chains, A and B, linked by multiple disulfide bridges, both within the A chain and between the A and B chains.
Very specific links.
Very specific. The complexity of these bonds meant that early attempts at direct chemical synthesis of the mature insulin molecule yielded very poor results. The cell needed a smarter way.
So the pre-prohormone solved that problem. How?
Yes. The reason became clear with the discovery that insulin is synthesized as a pre-prohormone, which is essentially a blueprint for many peptides processed from larger precursors. This pre-prohormone contains a hydrophobic pre- or leader sequence at its beginning.
The zip code you mentioned.
Exactly, like a zip code. This sequence acts like a zip code, directing the molecule into the endoplasmic reticulum, where it begins its folding. Once inside, this leader sequence is promptly removed.
And then it becomes proinsulin. What does that do?
Right. This removal results in the proinsulin molecule. Crucially, proinsulin provides the necessary shape and folding pathway for the proper and efficient formation of those complex disulfide bridges. It holds the A and B chains in the right orientation. Inside the cell, typically in the pancreatic beta cells, the proinsulin molecule undergoes a series of highly specific enzymatic cleavages at precise sites.
Cutting out the middle part.
Essentially, yes. These enzymatic cuts remove the connecting peptide, or C-peptide, resulting in the formation of equimolar amounts of mature, active insulin and the C-peptide. Both are secreted, and C-peptide levels are often used clinically as a direct indicator of how much insulin your body is making.
Another complex processing example is parathyroid hormone, or PTH, a key regulator of calcium. How is it synthesized and regulated? It also starts big.
Yes, parathyroid hormone PTH is initially synthesized as an even larger precursor. The primary gene product is called pre-pro-PH. This pre-pro-PH has an additional amino acid extension, a leader or signal sequence, which guides it into the cellular machinery for processing.
Same leader sequence idea.
Similar concept, yes. This signal sequence is then cleaved to yield pro-PTH, which is the immediate precursor to the mature hormone. Pro-PTH differs from the native 84 amino acid PTH by having a small, basic hexapeptide amino terminal extension that gets removed in the final steps.
So the mature hormone is 84 amino acids long. Which part is functionally active? Is the whole thing needed?
Actually, no. The full biological activity of PTH actually resides within its first 34 amino acids. This means that even a shorter fragment, PTH 134, is sufficient for all its biological functions, and a specific region within that fragment, around 2534, is primarily responsible for binding to the PTH receptor.
How is the synthesis and secretion of PTH regulated, especially by calcium? It's very sensitive.
Extremely sensitive. The biosynthesis and subsequent secretion of PTH are exquisitely regulated by the plasma ionized calcium, Ca2+, concentration through a complex process. An acute decrease in Ca2+ levels triggers a marked increase in PTH production and secretion. However, what's truly fascinating is that a significant amount, as much as 80% to 90% of the pro-PTH synthesized in the parathyroid cells, is actually rapidly degraded before it can be processed into intact PTH and secreted.
So degradation is a key control point. That seems counterintuitive. Why would the body waste so much of the hormone it just made?
It's not waste, it's brilliant regulation. This rate of degradation is inversely regulated by calcium. It decreases when Ca2+ concentrations are low, allowing more PTH to be secreted, and it increases when Ca2+ concentrations are high, efficiently reducing PTH output.
So high calcium tells it to destroy the precursor.
Essentially, yes. This precise control is mediated by a specific calcium-sensing receptor located on the surface of the parathyroid cell. This rapid calcium-dependent degradation acts as a powerful immediate brake or accelerator on PTH availability, allowing for very fine-tuned calcium homeostasis without relying solely on changing synthesis rates.
Another powerful hormone derived from a large precursor is angiotensin II, critical for blood pressure regulation. This one has a cascade involved, multiple steps outside the original cell.
Indeed. The renin-angiotensin system is central to regulating blood pressure and electrolyte metabolism, largely through the production of aldosterone. The primary active hormone in this system is angiotensin II, a small octopeptide. It's not made directly, but rather from angiotensinogen, a large protein produced in the liver.
So the liver makes the big precursor. And renin is the key enzyme that kicks off the process, right? From the kidney.
Yes. Renin is an enzyme produced by specialized cells in the kidney, the juxtaglomerular cells, which are highly sensitive to changes in blood pressure and salt levels. So any factors that decrease fluid volume, such as dehydration, decreased blood pressure, or fluid blood loss, or decreased salt concentration, will stimulate renin release.
The kidney senses low pressure and releases renin.
Exactly. Renin acts specifically on its substrate, angiotensinogen, clipping off a piece to produce the decay peptide, angiotensin I.
So angiotensin I is made, but it's angiotensin II that's the active one. What converts it? Where does that happen?
Angiotensin I is then converted to angiotensin II by angiotensin-converting enzyme, ACE. ACE is an enzyme found abundantly in the lungs and on the inner lining of blood vessels, the endothelium. It removes two amino acids from angiotensin I to form the active octopeptide angiotensin II.
And that conversion isn't the slow step.
Generally, no. The renin step is usually considered the rate-limiting step. The ACE conversion is relatively rapid.
And this is where ACE inhibitors come in for treating high blood pressure, isn't it? Blocking that conversion.
Precisely. Various compounds act as competitive inhibitors of ACE, effectively blocking the formation of angiotensin II. These ACE inhibitors are widely used to treat renin-dependent hypertension by preventing the formation of this powerful blood pressure-raising hormone.
What are angiotensin II's main effects once it's formed? It raises blood pressure, but how?
Angiotensin II is a very potent vasoactive substance. Its primary effect is to increase blood pressure by causing significant constriction of your arterioles.
Squeezes the blood vessels.
Yes. Beyond that, it also inhibits further renin release from the kidneys, a negative feedback loop, and is a powerful stimulator of aldosterone production from the adrenal cortex.
Which makes you retain salt and water.
Exactly. This stimulation of aldosterone then leads to increased sodium retention, expanded fluid volume, and ultimately a further increase in blood pressure. It's a multi-pronged approach to raising pressure.
How does angiotensin II exert its effects at the cellular level? Does it use second messengers like CAMP?
Interestingly, angiotensin II binds to specific receptors on cells in the adrenal cortex glomerulosa. But unlike many peptide hormones, this hormone receptor interaction does not activate adenylyl cyclase, and therefore, CAMP does not appear to be the primary messenger here.
So what does it use? Calcium again?
It seems so. Angiotensin II's actions, such as stimulating steroid hormone production, like aldosterone, likely involve rapid changes in the concentration of intracellular calcium and the metabolism of phospholipids, similar to other calcium and phosphoinositide-mediated signaling pathways we discussed earlier.
The pro-opiomelanocortin, or POMC peptide family, seems like an incredibly versatile precursor, generating a whole range of hormones and even neurotransmitters. This is a masterclass in making multiple tools from one blueprint.
It is a remarkable example of differential processing and biological efficiency. The POMC family consists of peptides that function both as hormones, such as adrenocorticotropic hormone, ACTH, lipotropin, LPH, and melanocyte-stimulating hormone, MSH.
ACTH we know from the adrenals.
Exactly. And others that serve as neurotransmitters or neuromodulators, like the endorphins, are natural pain relievers.
Wow, hormones and brain chemicals from the same starting point.
Yes. The initial precursor is a large protein of 267 amino acids, and it undergoes differential processing depending on the tissue, yielding a diverse array of active molecules.
Where is the POMC gene expressed, and how is it processed differently in different parts of the body? Mainly the pituitary.
The POMC gene is primarily expressed in the anterior and intermediate lobes of the pituitary gland. While the intermediate lobe is small or rudimentary in adult humans, it's very active in fetuses and pregnant women and in many animal species. Interestingly, POMC or related products are also found in several other tissues, including the brain, placenta, gastrointestinal tract, reproductive tract, lung, and even lymphocytes.
That was quite widespread. And the human POMC protein is processed into different polypeptide groups depending where it's made.
Yes. The processing begins with the removal of a signal peptide, generating the main POMC protein. From this precursor, 3 main polypeptide groups are generated via specific enzymatic cuts. First, ACTH, adrenocorticotropic hormone itself, can be further cleaved to give rise to IMSH and a related peptide called CLEP.
Okay.
Second, lipotropin, LEPH, can yield POMSH and 2 forms of endorphins, POBPs, which are potent opioid peptide.
The endorphin.
Yes, and third, a large N-terminal polypeptide, NPP, generates several forms of POMSH.
And this diversity comes from specific cleavage sites along the molecule, like dotted lines for the scissors.
Exactly. This remarkable diversity of products is generated via specific proteolysis enzymatic cutting at the locations of multiple dibasic amino acid clusters, like lysarg or arg-arg, along the length of the POMC molecule. Think of these as specific scissor points along the protein chain, recognized by cellular enzymes called prohormone convertases.
Yeah, there are other modifications too.
Yes. Furthermore, many of these small polypeptide hormones undergo extensive additional and tissue-specific modifications, such as adding phosphate groups, phosphorylation, acetyl groups, acetylation, or sugar molecules, glycosylation, or even amidation at the C-terminus. These modifications can significantly affect their biological activities and stability.
Amazing complexity. Any clinical relevance here?
Yes. Clinically, it's worth noting that mutations in the receptor for OR-MSH, one of these POMC-derived peptides, have been linked to a common early-onset form of obesity, highlighting the critical role of these peptides in energy balance and appetite regulation. Hashtag diversity in hormone storage and secretion.
We've covered hormone synthesis in incredible detail. Now let's look at how hormones are stored and secreted, which also shows remarkable diversity, reflecting their different chemical natures and physiological roles. It's not all stored the same way.
Not at all. There's a wide spectrum. For steroid hormones and 1025-OH2D3, they are synthesized in their final active form, and crucially, they are secreted immediately as they are made.
Made and shipped right away.
Exactly. This means there is no significant intracellular reservoir of these hormones. Their production rate directly determines their secretion rate. They are made on demand.
So their supply is constantly being refreshed based on need. What about catecholamines? We said they were stored in granules.
Catecholamines, like steroids, are also synthesized in their active form. However, unlike steroids, they are stored in granules within the chromaffin cells of the adrenal medulla. In response to appropriate neural stimulation, these granules are released from the cell through exocytosis, releasing the catecholamines into the circulation.
And they have a decent reserve.
Yes, these cells maintain a several-hour reserve supply of catecholamines, allowing for a rapid, sustained burst when needed during stress or excitement.
And PTH also has storage vesicles, but that unique regulatory mechanism involving degradation.
Yes, PTH exists in storage vesicles. What's unique about PTH, as we discussed, is that as much as 80% to 90% of the pro-PTH synthesized is actually degraded before it even enters this final storage compartment. This rapid degradation is particularly pronounced when calcium, Ci2+ levels, are high in the parathyroid cell.
So storage is linked to immediate breakdown if calcium's high.
Right. PTH is primarily secreted when Ca2+ levels are low in these cells, and they typically contain a several-hour supply of the hormone. This pre-secretory degradation is a very sensitive fine-tuning mechanism for calcium homeostasis.
Insulin is a hormone we definitely think of as being stored or ready for a meal. How much backup does it have?
Absolutely. The human pancreas secretes approximately 40 to 50 units of insulin daily, but this represents only about 15 to 20% of the hormone that is stored within the beta cells. Insulin and the C-peptide are normally secreted in equimolar amounts. Stimuli like glucose strongly provoke insulin secretion, and this also triggers the processing of proinsulin to insulin as an essential part of the secretory response. Beta cells maintain a substantial day's supply of insulin.
Days. Ready to respond to blood glucose fluctuations.
Exactly.
And the thyroid hormones have an even larger storage capacity, acting like a long-term reserve. The week's worth in colloid.
Indeed. T3 and T4 represent the most exaggerated example of a pro-hormone storage system. A several-week supply of T3 and T4 exists within the thyroglobulin molecule, which is stored as colloid in the lumen of the thyroid follicles. These hormones are then released upon stimulation by TSH. This means a very large precursor molecule must first be synthesized and then subsequently degraded to supply just a few molecules of the active hormones T4 and T3.
Such an elaborate system.
This extensive storage capacity provides a crucial buffer against variations in dietary iodine intake or changes in metabolic demand, ensuring a steady supply even if intake fluctuates.
We've covered synthesis and storage. Now let's talk about delivery. How are hormones transported in the plasma, and why is this so critical for their function? It's not as simple as just dissolving in the blood for all of them.
Not at all. This is a vital aspect, particularly for class I hormones. Hashtag A. The need for transport proteins. Class I hormones. Those fat-soluble ones like steroids and thyroid hormones. Due to their hydrophobic chemical nature, meaning they don't mix well with water, these hormones are not very soluble in the watery environment of plasma. This presents a significant challenge for their efficient circulation throughout the body to reach distant target cells.
So specialized plasma transport proteins act as their personal vehicles, solving the solubility problem, like chauffeurs.
Precisely. These specialized plasma transport proteins serve several crucial purposes. First, they effectively circumvent the solubility problem, enabling the hydrophobic hormones to be delivered efficiently to their distant target cells. Second, they provide a circulating reservoir of the hormone, which can be quite substantial, especially in the case of thyroid hormones.
The backup supply in the blood.
Exactly. This reservoir acts as a buffer against sudden changes in production. Third, when hormones are bound to these transport proteins, they are generally protected from rapid metabolism and degradation by enzymes, thereby prolonging their plasma half-life.
So they last longer because they're protected by the carrier.
Right.
And it's only the free hormone that's biologically active, correct? It's not about how much is in the reservoir, but how much is available to act now.
Yes, that's a fundamental principle. Only the free form of a hormone is biologically active and capable of diffusing out of the bloodstream to interact with its receptors on or within target cells. The binding affinity of a given hormone to its transporter significantly influences the bound versus free ratio. In general, the concentration of free hormone in plasma is exceedingly low, typically ranging from femtomolar to nanomolar levels.
Which again highlights why those receptors need to be so sensitive.
Exactly. Hashtag be hydrophilic hormones.
So if hydrophobic hormones need transporters, what about the hydrophilic ones, the water-soluble ones, like most peptide hormones and catecholamines? Do they need help?
Generally, no. The hydrophilic hormones, which generally belong to class 2 and have a peptide or protein structure, are freely soluble in plasma. Therefore, they do not require transport proteins. Hormones such as insulin, growth hormone, GH, ACTH, and TSH circulate in their free, active form.
You just swim freely in the blood.
Pretty much. As a consequence of being unprotected by binding proteins, they typically have very short plasma half-lives, often measured in minutes.
Quick on, quick off.
Meaning their effects are usually rapid and transient.
Is there an exception to this rule? A peptide that uses a carrier?
There is one notable exception that proves the rule. Insulin-like growth factor I, IGF-I, which, despite being a peptide hormone, is transported bound to members of a family of specific binding proteins, IGF-BPs.
Oh, interesting.
This highlights the complexity and nuances within biological systems, where exceptions often reveal deeper layers of regulatory control. Hashtag see specific plasma transport proteins.
Let's delve into some specific plasma transport proteins, starting with those for thyroid hormones. They have quite a large reserve in the blood, bound up.
Thyroid hormones, T4 and T3, are indeed extensively transported by binding proteins. A significant portion, 1/2 to 2/3 of the total T4 and T3 in the body, exists in an extrathyroidal reservoir, mostly circulating in a bound form. The primary binding protein is thyroxine-binding globulin, TBG. TBG is a glycoprotein that, under normal physiological conditions, non-covalently binds nearly all of the T4 and T3 in plasma.
And is there a difference in its affinity for T4 versus T3, and what's the implication for their half-lives?
Yes, TBG binds T4 with a greater affinity than it binds T3. It holds onto T4 more tightly. This difference directly influences their plasma half-lives. The plasma half-life of T4 is correspondingly 4 to 5 times longer than that of T3.
So T4 circulates longer because it's bound tighter.
Exactly. Despite the large difference in total amount circulating, the small unbound free fraction of T3 approximates that of free T4. Given that T3 is intrinsically much more active than T4, most of the biological activity of thyroid hormones is actually attributed to T3, often after T4 is converted to T3 in target tissues.
So the free amounts are similar, but T3 packs more punch.
That's a good way to put it. And TBG is also highly specific. It does not bind any other significant hormones.
What about glucocorticoids like cortisol, our main stress hormone? Do they have a specific transporter, or do they just float free?
No, they have help too. Glucocorticoids are also transported. Hydrocortisone, or cortisol, circulates in both protein-bound and free forms. The main plasma-binding protein for cortisol is an alpha-globulin called transcortin, or more commonly corticosteroid-binding globulin, CBG.
CBG for cortisol.
Right. CBG is produced in the liver, and its synthesis, similar to TBG, is increased by estrogens. At normal plasma cortisol levels, CBG binds most of the hormone, although smaller amounts of cortisol are also bound to albumin, another general plasma protein.
How does binding affinity relate to their half-life here? Is tighter binding longer lasting?
Yes, same principle. The affinity of binding directly helps determine the biological half-lives of various glucocorticoids. Cortisol binds tightly to CBG and consequently has a plasma half-life of 1.5 to 2 hours. In contrast, corticosterone, a less potent glucocorticoid, binds less tightly to CBG and has a shorter half-life of less than 1 hour.
Okay. And again, only the free stuff is active.
Exactly. Only the unbound or free cortisol, which constitutes approximately 8% of the total, represents the biologically active fraction. It's also worth noting that CBG binding isn't exclusive to cortisol. Related steroids like deoxycorticosterone and progesterone can also bind to it. However, aldosterone, the most potent natural mineralocorticoid, does not have a specific plasma transport protein. It mostly travels bound loosely to albumin or free.
Interesting. Aldosterone goes it alone mostly. And finally, for gonadal we have sex hormone-binding globulin, SHBG, which sounds like it plays a crucial role in balancing male and female hormones.
It does. Most mammals, including humans, possess a plasma protein that binds testosterone with high specificity, relatively high affinity, and limited capacity. This protein is commonly referred to as sex hormone binding globulin, SHBG, or sometimes testosterone estrogen binding globulin, TBG.
SHBG. And it binds both.
It binds both testosterone and estradiol, yes. Like other major binding proteins, SHBG is produced in the liver. Its production is increased by estrogens, which is why women typically have twice the serum concentration of SHBG as men, as well as certain liver diseases and hyperthyroidism. Conversely, its production is decreased by androgens, advancing age, and hypothyroidism.
So its levels can really fluctuate depending on conditions. How much testosterone is actually free and active, considering SHBG? Is it also a small percentage?
Yes, a very significant amount is bound. SHBG and albumin together bind 97% to 99% of circulating testosterone. This means only a very small fraction, 1-3% of the hormone in circulation, is in the free, biologically active form.
Wow.
Yeah. The primary function of SHBG may be to precisely restrict the free concentration of testosterone in the serum, thereby modulating its bioavailability and ensuring tight control over its effects.
And there are differences in binding affinity for various steroids, right? It seems complicated who binds to what and how tightly.
Yes, very important differences. Testosterone binds to SHBG with higher affinity than does estradiol. Therefore, a change in the level of SHBG will cause a greater change in the free testosterone level than in the free estradiol level.
So changes in SHBG affect free testosterone more.
Disproportionately affect male hormone activity, yes. While estrogens are bound to SHBG, progestins are primarily bound to CBG. SHBG binds estradiol about 5 times less avidly than it binds testosterone or DHT, and progesterone and cortisol have little affinity for SHBG. Conversely, progesterone and cortisol bind with nearly equal affinity to CBG, which in turn has little affinity for estradiol and even less for testosterone, DHT, or estrone.
Okay, so it's a complex web of who binds where.
It ensures the right hormone gets to the right place in the right amount, or rather, the right free amount.
So these binding proteins really act as a buffer, preventing sudden fluctuations in hormone levels and ensuring steady delivery over time.
Exactly. These binding proteins provide a crucial circulating reservoir of hormones. Because of their relatively large binding capacity, they effectively buffer against sudden changes in plasma levels, ensuring a more stable and controlled hormonal environment. Furthermore, the rate at which these steroids are cleared from the body is inversely related to how tightly they bind to SHBG.
So tighter binding means slower clearance.
Generally, yes. For example, estrone is cleared more rapidly than estradiol, which in turn is cleared more rapidly than testosterone or DHT, reflecting their differential binding affinity to SHBG. It's a system designed for precision, stability, and selective action. #tag outro.
What an incredible journey we've taken through the endocrine system. I mean, from the surprising definitions of hormones expanding beyond just distant messengers to local communicators, to the critical concept of the target cell and the myriad factors that truly determine its unique response.
So much complexity in just getting the signal there and having it heard correctly.
Absolutely. We've unpacked the central role of hormone receptors, exploring their elegant dual functional domains and the 2 general ways they transduce signals, whether from the cell surface or within. It's like discovering the cell's secret handshake for every incoming message.
We truly delved into the profound chemical diversity of hormones, from those sculpted from cholesterol, like the steroids and vitamin D, to those derived from tyrosine, like catecholamines and thyroid hormones.
And the intricate processing of large peptide precursors, insulin, PTH, POMC, making multiple active forms from one starting point.
Understanding the step-by-step synthesis pathways truly lays bare the elegance and precise control mechanisms that govern these vital messengers. It's a masterclass in biochemical engineering that happens inside you every second.
And let's not forget the fascinating diversity in how hormones are stored and secreted, made on demand, stored for hours, days, or even weeks in vast reserves like the thyroid colloid.
Different strategies for different needs.
Finally, we explored the crucial role of plasma transport proteins, which don't just deliver hydrophobic hormones, but also buffer their levels and prolong their active lives in the bloodstream. This deep dive has truly revealed how each minute detail contributes to the overall communication network that allows us to adapt, thrive, and maintain balance against an ever-changing internal and external world.
We hope this deep dive has given you, our listener, a truly foundational understanding, the kind that empowers you to look at your own body's processes with new eyes and, a deeper appreciation for the intricate biochemistry behind it all.
Yeah, seeing the interconnected genius in every function.
Absolutely.
So, if the intricate dance of hormones allows multicellular organisms to adapt to a constantly changing environment, What does this tell us about the ultimate adaptability of life itself? It makes you wonder.
It does. And how might a deeper understanding of these fundamental biochemical principles unlock future treatments or even enhance human potential in ways we can only begin to imagine?
Think about it. What other biological systems in the body operate with similar complexity and multi-level regulation? Maybe the immune system or neurological networks.
Great examples. Perhaps this deep dive has sparked new questions in your own curiosity, urging you to explore further the hidden biochemical wonders that make life possible.
Until next time, keep diving deep.